Hello, this is Dr. Paningrai. Welcome to my biology classes for NEET exam. Today I will be talking about the antigen-antibody reaction. So before going through this antibody and antigen reaction, you have to know what is antigen. Antigen, it may be any foreign materials which stimulate the immune response in our body. It may be protein or it may be polysaccharides. Lipids along with protein, it may act as antigen. Such antigen may be the different pollens, may be the blood of other organisms which also activate the immune system. And the antibody, as you know, antibody is the protein which secreted from the plasma cell in antigenic stimulation. So these are the Y-shaped general structure of antibody. As you know, this portion is known as the FAB, the fragment of antigen binding, and this is the fragment of crystallizable. And such portion, the top of the Y-shaped region, as you know, this is the amino terminal and this is the C terminal. These are the amino terminal. And you know, this is the heavy chain and this is the light chain. Such heavy chain and light chain towards the amino terminal region, this is known as the variable region. So the variable region of light chain and heavy chain which forms the antigen binding site, this is known as the antigen binding site. Antigen binding site. On this site, the antigen binds with the antibody. Suppose that this is the antigen having the very specific region, and such region actually recognized by antibody, and this region is known as the antigenic determinant site. This is known as the antigenic determinant site, or it also known as the epitope. This is known as the epitope. So such portion actually recognized by the antibody, and such antibody binds or trap the antigen by the binding with the antigen, and which forms, suppose that this is the antigen and this is the antibody, which forms antigen antibody complex. So the binding of antibody with antigen which forms the complex known as the antigen antibody complex. Such complex or such antigen actually destroyed or inactivate or neutralized by different effector mechanism, and these may be several types. So the complex of antigen and antibody which is destroyed or neutralized or inactivate by different effector mechanism. So one may be the agglutination. Agglutination is the one effector mechanism by which the antibody and antigen complex inactivate the antigen. Suppose that this is the antigen and this is the antibody. So the binding of antigen antibody or the complex, such complex precipitate the antigen. As a result, antigens become inactive or harmless. This is known as the agglutination. The another effector mechanism, it is known as the opsonization. Opsonization is the process if the antigen, when it binds with the antibody, now such antigen easily recognized by the phagocytic cell. Suppose that this may be the phagocytic cell. So after the binding with antibody, the antigen easily recognized by the phagocytic cell, and the phagocytic cell engulf such antigen and destroyed the antigen. Another effector mechanism may be the neutralization. So by the binding of antibody with antigen, such antigen may inactivate or such antigen may neutral. This is known as the neutralization. The effect, the harmful effect of the antigen become harmless. That's why it is known as the neutralization. Another effector mechanism may be the cell lysis by complement system, complement protein or system. So the binding or the formation of antibody and antigen complex which also activate the complement system. Such complement system may lysis the cell or lysis the antigen. So this is the total mechanism of the antigen antibody reaction. I hope you understand better. If you like, then click on my next video to get the more video like this. Thank you for clicking.